You know, walking around in autumn, seeing those leaves turn brilliant colors and then just fall to the ground. It feels like things are just giving up, right? Like decay taking over, a slow fade.
It certainly looks that way on the surface, but when you dig a little deeper, inside the plant, what's happening is anything but passive. It's one of the
Yeah.
most intricate, carefully orchestrated biological programs there is.
A program for self-destruction.
Essentially, yes.
Yeah.
In plants, the death of cells, organs, or even the entire organism isn't usually random decay. It's often an active, genetically controlled process. It's essential for their life cycle, for growth, and for getting ready for the next generation.
Okay, that's fascinating. This deep dive is all about peeling back the layers on that plant senescence and cell death. We're drawing our insights today from Chapter 22 of Plant Physiology and Development, looking at the sophisticated mechanisms, stages, and how it's all regulated.
Right. And to get started, let's clarify a couple of terms because they're key to understanding this. Senescence is the active, energy-dependent, genetically programmed process that leads to the decline and eventual death of specific plant parts, or the whole plant. It's plant aging, basically.
Okay, plan aging.
And necrosis, on the other hand, is just passive death, usually caused by injury, extreme stress, or toxins. Think of like frost damage on a leaf. That's necrosis.
So senescence is the plant choosing to shut something down. Necrosis is something just breaking.
That's a good way to put it, yeah.
Yeah.
And then there's abscission, which is the controlled shedding of organs like leaves, flowers, or fruits. It often happens after senescence has prepared the organ for removal. It's the final controlled detachment step.
So the plant doesn't just let things rot, it plans their departure and sometimes even uses cell death to, well, to manage it.
Exactly. And the fundamental cellular engine driving a lot of this is programmed cell death, or PCD. This is a cell's ability to essentially commit suicide in a controlled way. It's vital for normal plant development like shaping tissues or creating structures, and also for defense against threats.
So PCD is the smallest scale, the individual cell doing this.
Right. And you can see PCD happening at different levels in a plant. There's single-cell PCD, which underlies processes like forming those water-transporting xylem vessels. Then there's organ senescence, like a leaf dying off, where many cells are undergoing PCD in a coordinated way.
Okay.
And finally, whole plant senescence, where the entire organism undergoes a programmed decline after reproduction, which also involves widespread cellular PCD. So PCD is really the fundamental building block for programmed plant deaths at any scale.
How does plant cell suicide work? Is it like apoptosis in animals?
There are parallels, definitely, but key differences too. In animals, apoptosis involves this neat packaging of cell contents into little bubbles for cleanup by other cells. Plants don't really do that, mainly because of the rigid cell wall.
The wall gets in the way.
It does. Their process is often more of a self-digestion, called autolysis. They use specific enzymes, including proteases that are functionally similar to animal caspases, but they aren't actually the same proteins. And the way their DNA breaks down can look different too.
Okay, so the goal is similar controlled breakdown, but the cellular mechanics are distinct. Are there different types of plant PCD based on how it happens?
Yes. Our source highlight two main ones based on how the cell breaks down. There's vacuolar type PCD, which is common during normal development. The cell's large central vacuole, which is full of digestive enzymes, actually swells up and bursts,
Whoa.
releasing those enzymes to break down everything inside the cell. Think of creating those hollow tubes for water transport in xylem vessels that uses this type of PCD. It's also involved in sculpting tissues and, importantly, developmental leaf senescence.
So building and shaping uses this sort of internal explosion. What's the other type?
That's hypersensitive response type, or HR-type PCD. This is primarily a defense strategy, often triggered by a pathogen attack. It's almost the opposite of vacuolar PCD here. The vacuole loses water rapidly, the cell shrinks and pulls away from the wall.
Shrinks instead of swells.
Exactly. This happens very quickly in cells right around an infection site, creating a dead zone that effectively walls off the pathogen and prevents it from spreading further. The plant basically sacrifices a few cells to save the whole organism.
Ah, one for construction and development, one for rapid defense. Makes sense. Now, you mentioned a process that sounds central to breaking things down. Autophagy.
Yes, autophagy, which literally means self-eating. This is a crucial cellular recycling system. It helps clear out damaged proteins and organelles, old cellular parts, and it's absolutely essential for mobilizing resources when the cell is stressed, especially by nutrient limitation. It's like the cell's internal cleanup and salvage crew.
How does this self-eating actually work? Can you walk us through it?
Sure. The main process we talk about is macrotophagy. It starts with a membrane structure forming, often originating from the endoplasmic reticulum called a phagophore. Think of it as like a tiny crescent-shaped membrane starting to grow within the cytoplasm.
Okay.
This membrane then expands, kind of reaching out to engulf whatever needs to be recycled, maybe a damaged mitochondrion or a clump of old proteins. It then closes up, forming a double membrane bubble called an autophagosome, with the cargo trapped safely inside.
Like wrapping up the cellular trash in a bag.
Exactly, a very specialized bag. This autophagosome then travels through the cytoplasm to the vacuole, that big central compartment. It fuses with the vacuole membrane.
And dumps the contents.
Pretty much. It releases its inner membrane, and the contents now called an autophagic body inside the vacuole. And the vacuole, remember, is full of digestive enzymes. These enzymes then break everything down into basic building blocks, amino acids, sugars, fatty acids, which are then transported back out into the cytoplasm for the cell to reuse. It's incredibly efficient recycling.
Wow, and this is all controlled by specific cellular machinery, I assume?
Absolutely. There's a whole set of conserved genes called ATG genes for autophagy related, and the proteins they code for make up the core autophagy machinery. They assemble at specific sites called the PAS to orchestrate the formation of that phagophore and autophagosome. It's a complex molecular machine.
Is there a master switch for when this recycling kicks in? Like, how does the cell know it's needed?
A key regulator is a protein kinase called TOR, target of rapamycin. TOR acts like a brake on autophagy. When nutrients are plentiful and the plant is growing well, TORS is active and it suppresses autophagy. Things are good, no need for intense recycling.
Right.
But when the plant experiences nutrient stress, like nitrogen starvation or other stresses, TOR activity gets inhibited. This takes the brake off, and autophagy ramps up significantly, allowing the cell to recycle its own components to survive the tough times.
So it's directly linked to the cell's perception of its nutritional status. What's autophagy's role in the bigger picture of plant life and death, then? It sounds like survival.
It has a dual role, really. First, it's absolutely essential for cell homeostasis, just maintaining health and preventing premature decline by constantly cleaning house, getting rid of damaged stuff. Experiments with Arabidopsis plants, where core autophagy genes were mutated, showed they senesce much faster and grow poorly.
So without it, they age faster.
Yes, they can't keep up with the cellular maintenance. It keeps the cell alive longer and healthier under normal conditions. But, and this is the interesting part, it also plays a role in pathological processes. It actually contributes to executing some programmed death pathways, like that HR-type defense response we discussed earlier.
So it can be a tool for survival or a tool for programmed death, depending on the situation.
Exactly. It depends on the context and the signals the cell is receiving.
That dynamic role is fascinating. Okay, let's move up in scale a bit. What about the really familiar process of leaf senescence, the autumn colors and falling leaves?
Right. Leaf senescence. That programmed aging of an individual leaf. And it's not just for deciduous trees dropping leaves in fall. All leaves, even on evergreen plants, undergo senescence eventually. Think about that incredible plant Welwitschia mirabilis down in the Namib Desert. It only has 2 leaves that grow continuously from the base their whole lives, while the tips are constantly senescing and dying off.
A constantly aging and growing leaf. That's wild. Seriously.
It really is. But the key benefit of leaf senescence, especially in deciduous plants or just older leaves on any plant, is nutrient remobilization. The plant doesn't just discard the leaf's valuable components. Proteins, carbohydrates, nucleic acids are systematically broken down.
Okay.
And the resulting amino acids, sugars, minerals, particularly nitrogen, which is often a limiting nutrient, are transported out of the senescing leaf via the phloem and into growing parts of the plant, or maybe into developing seeds and fruits.
So the plant is essentially reclaiming all the valuable resources before letting the structure go.
Precisely. It's a highly efficient strategy for recycling nutrients. And this happens as part of the leaf's normal developmental plan, even when environmental conditions are perfectly good. It's about maximizing resource use over the plant's life.
You mentioned developmental age earlier. Does a leaf's actual age, like how many days it's been alive, matter as much as where it is in its overall developmental plan?
That's a really critical point. A leaf's developmental stage and the signals it receives are often much more important than its chronological age. There's a classic experiment from way back in 1909 by Ernst Stahl. He cut a disc out of a green mock orange leaf and kept it floating on a nutrient solution in the lab.
Okay.
The intact leaf still on the plant turned yellow and senesced normally in the fall. But the cut disc, it stayed green.
Because it was cut off from the rest of the plant's signals.
Exactly. It wasn't receiving the internal signals hormonal changes, nutrient shifts from the rest of the plant, nor the external environmental cues like shorter days and cooler temperatures that were triggering senescence in the leaf still attached to the tree. This clearly showed that senescence is actively triggered by signals. It's not just a passive clock running out.
Got it. So signals are key. What kind of patterns do we see in leaf senescence? Is it always the same?
Now, you see a few main types. There's sequential senescence, which is common in many plants. The older leaves at the bottom of the plant senesce and die first, and the senescence progresses upwards towards the younger leaves.
Right, you see that on things like tomato plants.
Yep. Then there's seasonal senescence, like the classic autumn color change, where basically all the leaves on a deciduous plant senesce more or less together, typically in response to environmental cues like day length and temperature. Both of those are considered forms of developmental senescence.
And the third?
Then there's stress-induced senescence. This is premature senescence, often localized to certain leaves or parts of leaves, triggered by unfavorable conditions like drought, extreme heat, lack of nutrients, or disease attack.
Okay. Are there specific stages you can identify within developmental leaf senescence? Like, how does it unfold?
Yes, generally you can break it down into 3 phases. First, there's the initiation phase. This is where the signaling really begins, photosynthesis starts to decline noticeably, and the leaf begins to transition from being a nutrient sink, importing sugars, to a nutrient source, exporting sugars and other breakdown products.
So it flips its role.
Right. Then comes the degenerative phase. This is the main event, really. You get intensive breakdown autolysis of cellular components like proteins and chlorophyll. Nutrients are actively exported out of the leaf, and importantly, the abscission layer at the base of the leaf starts to form during this phase.
Getting ready for detachment.
Exactly. And finally, the terminal phase. Breakdown is essentially complete. Cell separation occurs in that abscission zone, and the leaf detaches or abscisses from the plant.
What's one of the very earliest things you'd actually see happening inside the cell as this process kicks off?
One of the most prominent and earliest visible changes is the degradation of chloroplasts. Remember, chloroplasts contain the vast majority of the leaf's protein, primarily rubisco, the main enzyme for carbon fixation. So breaking them down is absolutely key for nutrient recycling, especially nitrogen.
And they change appearance.
Yes, they transform into structures called gerontoplasts. This transformation isn't just about breakdown. It's also vital for safely dismantling the chlorophyll and the associated light harvesting machinery.
Why safely? Isn't chlorophyll just a green pigment?
Chlorophyll, and especially some of its breakdown products, can be highly photoreactive. If they weren't broken down in a very controlled manner within the gerontoplast, they could absorb light energy and generate damaging reactive oxygen species, causing chaos inside the dying cell. So the structural changes, like the internal membrane stacks, the grana coming apart, and the formation of lipid droplets called plastoglobuli, are part of this controlled dismantling.
Okay, safe dismantling. What about other key organelles, like the nucleus or mitochondria?
They tend to hang around longer. The nucleus and mitochondria are generally needed to orchestrate the later stages of senescence and the export process, so they're usually degraded later in the terminal phase. Interestingly, the chloroplasts in the guard cells, those little cells that control the leaf pores or stomata, are often the very last ones to break down.
Huh. Where does all that chloroplast protein actually get broken down? Inside the chloroplast itself.
It's complex, and it happens in a few different locations. Some protein degradation does occur right there, via proteases located inside the chloroplast, especially early on. But a lot of the protein, particularly the abundant rubisco, seems to be moved outside the chloroplast before being degraded. This can happen in specialized protease-rich vacuoles called SAVs, or senescence-associated vacuoles, or perhaps other structures.
And autophagy plays a role here, too.
A major role, yes. While some components might be broken down piecemeal, experiments, particularly using dark-induced senescence and Arabidopsis, have shown that autophagy, that self-eating process, is required for the breakdown and recycling of entire chloroplasts. Leaves that had their autophagy machinery disabled couldn't properly degrade whole chloroplasts during senescence.
So autophagy clears out the whole organelle eventually. Is there a specific protein that's really critical for getting the chlorophyll part dismantled?
Yes, a very important protein is called stay green, or SGR. Chlorophyll doesn't just float around, it's tightly bound to proteins, especially in the light harvesting complexes, LHCs. SGR is required to somehow destabilize these chlorophyll protein complexes, essentially releasing the chlorophyll from the proteins. This allows the proteins to be broken down and recycled, and allows the chlorophyll itself to enter its specific breakdown pathway, avoiding toxicity.
And if SGR is missing?
Plants with mutations in the SGR gene can't efficiently detach the chlorophyll. So the leaves stay visibly green much longer during senescence, even though the underlying breakdown of proteins and loss of function is still happening. It's a cosmetic effect. In fact, Gregor Mendel's classic work with peas, one of the traits he studied, green versus yellow cotyledons, was actually due to a mutation in an SGR gene.
No way. That's a fantastic connection. It really highlights that staying green doesn't mean the leaf is still fully functional. It just means it hasn't managed to take out the chlorophyll trash properly.
Exactly. The functional senescence, the loss of photosynthetic capacity and nutrient export, is still happening. And this whole transition into senescence, as you might imagine, involves a massive shift in gene activity. Literally hundreds of genes, called senescence-associated genes, SAGs, are turned up, while others, senescence-downregulated genes, or SDGs, are turned off.
A huge genetic reprogramming event. How is all of this orchestrated? What controls which genes get turned on or off?
It's managed by a very complex regulatory network. Think of it like a web. It integrates signals from multiple sources. You've got internal signals, things like the levels of various plant hormones, sugar concentrations within the leaf, and the leaf's own developmental stage or age. And you have external signals from the environment, light duration and quality, temperature, water availability, nutrient status, pathogen attack.
All feeding into this network.
Right. These signals interact and converge through various signaling pathways involving things like reactive oxygen species, protein kinases, epigenetic changes, small RNAs, to ultimately regulate the activity of key transcription factors. And it's these transcription factors that bind to the DNA and control whether specific SAGs or SDGs are switched on or off.
Are there particular families of transcription factors that are known to be major players in controlling senescence?
Yes, our source highlights two major families that are particularly important in plants: the NAC family and the WRKY family. These are large families of transcription factors mostly unique to plants. A really clear and impactful example of the NAC family's importance comes from wheat, involving the NM genes.
NM, what does that do?
Yes, NM stands for no apical meristem, but these specific ones are involved in senescence. In wheat, the presence of a functional version, an allele, of the NME1 gene promotes earlier leaf senescence. And crucially, this earlier senescence drives a more efficient movement of nutrients, especially nitrogen, but also important minerals like iron and zinc from the senescing leaves into the developing grain.
So earlier leaf death in wheat actually helps make the grain more nutritious. That sounds counterintuitive initially.
It does, but it makes sense from the plant's perspective of provisioning the next generation, and the experimental evidence is striking. Researchers created bred wheat lines where they reduced the expression of the NM genes. What happened? The leaves stayed green longer, delaying senescence. But the resulting grain had significantly lower protein content and lower levels of iron and zinc. It's a powerful demonstration of how this genetically programmed senescence process is directly linked to grain quality and the nutritional value we rely on.
Wow, that's a really direct link between programmed death and agricultural value. What about the other family you mentioned, WRKY?
The WRKY family of transcription factors is also widely involved in plant responses, often linking stress signaling to developmental processes, including senescence. In the model plant Arabidopsis, for example, a specific factor called WRKY53 has been identified as a key positive regulator. If you knock out the WRKY53 gene, the plants show significantly delayed leaf senescence.
So it acts like an accelerator for senescence. What about other signaling molecules? We touched on ROS and sugars earlier.
Yes, they're definitely part of the network. Reactive oxygen species, ROS, like hydrogen peroxide H2O2, are interesting. While they can be toxic at high levels, causing damage, at lower levels they also act as important internal signaling molecules within the senescence program. They can trigger specific pathways and even induce the expression of certain SAGs, including transcription factors like WRKY53, linking oxidative signals to the core genetic controls.
So a bit of stress signal is actually part of the plan.
In a controlled way, yes. And sugars themselves can also act as signals. High sugar concentrations, especially when combined with low nitrogen levels, can be interpreted by the plant as a sign of resource imbalance, potentially triggering senescence pathways.
And hormones, of course. They seem to pop up everywhere in plant signaling. How do they fit into the senescence picture?
Absolutely central. Plant hormones play crucial, although often very complex and interconnected, roles in modulating senescence. It's important to note they often don't necessarily initiate the process that might be more linked to developmental age or strong environmental cues, but they definitely fine-tune its timing, its rate, and its progression once the leaf or organ reaches a certain competency to respond.
Mm.
And their pathways overlap and interact extensively. It's rarely just one hormone acting alone.
Okay, so who are the main players pushing things towards senescence, the accelerators?
Several hormones act as positive regulators, promoting senescence. Ethylene is a major one. It strongly accelerates senescence and is particularly vital for the process of abscission, the shedding part. Abscisic acid, ABA, generally enhances senescence, and its levels often increase under stress conditions like drought, providing a link between water stress and senescence. There's even a specific SAG regulated by ABA that affects stomatal closure in senescing leaves.
Interesting. Others?
Yes, jasmonic acid, GA, often associated with defense against insects and necrotrophic pathogens, also stimulates senescence. Its role might be even stronger in flower senescence. Brassinosteroids, BRs, which are involved in many aspects of growth and development, also generally accelerate senescence. And salicylic acid, SA, famous for its role in defense against biotrophic pathogens, also promotes senescence and induces various SAGs, including some WRKY factors.
Wow, quite a few pushing it forward. What about the ones that try to hold it back to keep things green and functioning?
The most well-known and perhaps universal repressors of senescence are the cytokinins. They strongly delay senescence. If you apply cytokinin to a small spot on a leaf that's starting to senesce, that spot will stay green, while the surrounding tissue yellows, the classic green island effect.
I've seen pictures of that.
Yeah. Cytokinin levels naturally tend to decline in mature leaves as they approach senescence, and maintaining higher cytokinin levels, either through external application or genetic modification, can dramatically delay the process. This is linked to their role in nutrient allocation. Cytokinin-rich areas tend to act as nutrient sinks, drawing resources towards them. There's a great experiment showing cytokinin treatment makes a cucumber cotyledon act as a strong sink.
So keeping cytokinin high keeps the leaf acting young, basically. Any others that hold back senescence?
Auxins, primarily known as growth hormones, also often act as negative regulators of senescence, particularly in leaves. High auxin levels tend to inhibit the expression of many SAGs, and gibberellins, GAs, another group of growth-promoting hormones, also generally repress or delay senescence. Their active levels also tend to decline with leaf age.
It really sounds like a complex hormonal balancing act, constantly integrating all these internal and external cues to decide when and how fast senescence proceeds.
It really is a dynamic interplay.
Yeah.
And that brings us logically to the final act for many leaves and other organs, abscission.
The shedding, the letting go.
Yes, that controlled shedding process. This happens at a specialized predetermined region called the abscission zone, usually located right at the base where the leaf stalk, petiole, flower stalk, or fruit stalk joins the main stem. This zone contains distinct layers of cells formed early in the organ's development. Some mutants, like the jointless tomato, actually lack this zone and can't shed their fruit properly.
So it's a specific anatomical feature. What happens physically there to make the organ detach?
Within the abscission zone, a specific layer called the separation layer becomes active. Cells in this layer start producing and secreting enzymes, particularly cellulases and polygalacturonases, that specifically target and break down the pectin and cellulose in the middle lamella, the glue holding the cell walls of adjacent cells together.
So they dissolve the connections between cells.
Exactly. This weakens the tissue dramatically. So eventually the weight of the organ, or wind, or maybe some cell expansion nearby, causes a clean break along that separation layer without tearing the underlying tissues on the stem. It's important to note this is primarily programmed cell separation, not necessarily widespread cell death right in that layer itself, although PTD often occurs in the senescing organ just before or during abscission.
And how is this separation process controlled? Hormones again, I bet.
You bet. It's largely regulated by the interplay between auxin and ethylene, acting specifically on the cells within the abscission zone. The model goes like this. A young, healthy leaf produces high levels of auxin, which is transported down the petiole. This high auxin flow actually suppresses the sensitivity of the abscission zone cells to ethylene, keeps them dormant.
So auxin protects against shedding.
Right. But as the leaf ages and begins to senesce, or if it experiences certain stresses, its auxin production declines significantly. At the same time, ethylene production often increases, both in the senescing leaf and potentially in the abscission zone itself. This combination, falling auxin and rising ethylene, makes the cells in the abscission zone become highly sensitive to the ethylene signal.
So ethylene flips the switch once auxin levels drop.
Precisely. Ethylene acts as the primary trigger for the abscission process once the zone has become sensitive. The ethylene signal then activates the genes responsible for producing those cell wall-degrading enzymes, cellulase, polyglycerol. These enzymes do their work, the cell connections loosen, and the organ drops off. You can see this dramatically treating birch trees with ethylene causes mass defoliation, while ethylene-insensitive mutants often fail to shed their leaves normally.
Okay, so we've gone from single cells dying programmatically to organs like leaves senescing and shedding. What about the death of the entire plant?
Right, that's whole plant senescence. This is most obvious and well-defined in monocarpic plants. These are plants that go through their entire life cycle, flower, set seed, and then the entire plant undergoes a programmed senescence and dies. This includes all annuals like corn or beans, biennials like carrots in their second year, and even some very long-lived but single-reproducing perennials like the century plant, agave, which might live for decades before one massive flowering event, or certain bamboo species where entire clonal populations flower synchronously across huge areas after many years and then die off together.
Wow, synchronous death after flowering. That's dramatic.
It is. The alternative is polycarpic plants like most trees, shrubs, and herbaceous perennials, which flower and reproduce multiple times over their lifespan and don't typically undergo whole plant program death after each reproductive cycle.
So in those monocarpic plants, the whole organism seems programmed for death right after it reproduces.
Yes, it's tightly linked to reproduction. And a key question for decades has been, what actually triggers this whole plant senescence? Is there a death signal sent out after flowering? The strongest evidence, though, points back to the developing reproductive structures themselves, the seeds and fruits.
The babies drain the parent.
Essentially, yes. Developing seeds and fruits are incredibly powerful metabolic sinks. They demand enormous amounts of resources, sugars, amino acids, minerals which are drawn away from the vegetative parts of the plant, leaves, stems, roots. There are classic experiments, for instance, with soybean, where if you continuously remove the flowers or young pods, preventing seed development, you can often keep the plant alive and vegetative much longer, sometimes indefinitely, preventing the onset of whole plant senescence.
So it really seems like the massive drain of resources allocated to reproduction is the primary trigger that flips the switch for the whole plant to decline.
That's the dominant model. The intense, overwhelming demand from these developing reproductive sinks dramatically alters the plant's overall source-sink balance. This likely leads to systemic changes in nutrient status, like the overall carbon-nitrogen ratio, and hormonal signals throughout the plant, which then activate the genetic programs for senescence in the vegetative tissues. It's probably not just simple starvation, as leaf carbohydrate levels can sometimes even increase during senescence, but rather a systemic signal of resource reallocation and completion of the reproductive mission.
What about long-lived perennials then, like those massive ancient trees? Do they just slowly age and decline over centuries? Does their growth just gradually slow down until they fade away?
That was certainly the traditional view for a long time. The idea was that as trees get very large and old, factors like the increasing difficulty of transporting water to the top, hydraulic limitation, or perhaps reduced photosynthetic efficiency in older leaves would cause the overall growth rate to slow down, leading to a gradual decline or senescence.
Makes intuitive sense.
It does. But surprisingly, recent, very large-scale studies that meticulously tracked carbon accumulation in individual trees across diverse forests and continents have provided strong counter-evidence.
Oh, what did these big studies actually find?
They found that for most tree species examined, the rate of total above-ground biomass accumulation, essentially how fast the tree is adding wood and growing in total mass, actually increases continuously as the tree gets larger and older.
Wait, hang on. Bigger, older trees are actually growing faster in total, adding more wood per year than younger, smaller ones.
Yes, that's what the data overwhelmingly suggests for the majority of species. While the photosynthetic efficiency per unit of leaf area might decline slightly in older trees, or growth rate relative to size might slow, the sheer total leaf area of a massive tree becomes enormous. And as the trunk diameter increases, the total leaf area it supports continues to increase substantially. This massive increase in total photosynthetic capacity, more leaves overall, more than compensates for any slight decrease in efficiency per leaf, leading to a faster absolute rate of carbon gain and biomass accumulation in the largest, oldest individuals.
That completely flips the script on how I thought about old trees. If they're growing faster than ever, potentially right up until they die, why do they eventually die? It doesn't sound like senescence in the sense of a slow, programmed aging decline.
Exactly. It strongly suggests that senescence and death in large, old trees isn't typically a gradual decline caused by physiological old age or running out of steam in the way we might think of animal aging. Instead, they seem to be operating at peak or even increasing growth capacity. Their eventual death is more likely triggered by external factors reaching a tipping point, perhaps mechanical failure under their own immense weight, susceptibility to windstorms, a severe drought, or an overwhelming attack by pests or pathogens. Or maybe complex internal failures not simply related to a slow decline.
So less fading away, more potentially catastrophic failure while still growing strong.
That seems to be a more accurate picture based on this recent evidence. Death in the giant ancient tree might not be the end of a slow, inevitable decline, but potentially a more abrupt, massive system failure hitting an organism that was, until that point, still highly productive and growing vigorously.
Wow, okay. This whole deep dive has completely reframed how I think about plants dying. It's really not passive decay at all, is it?
Not usually, no. From the carefully planned self-destruction of single cells that sculpts tissues or creates defensive barriers, to the strategic dismantling and recycling of resources in a senescing leaf before it's shed, right up to the complex life cycle decisions governing the fate of the whole plant and even challenging our assumptions about how ancient trees grow. It's all remarkably active and programmed.
Death is a strategy, almost.
In many ways, yes. It shows that death in the plant kingdom is often an essential, integrated part of life, a process carefully controlled by genetics, modulated by the environment, and orchestrated by hormones to ultimately benefit nutrient cycling within the ecosystem, defense against threats, and, crucially, the success of the next generation.
It definitely makes you look at a falling leaf, or even an old tree, with a whole new level of respect for the complex biology at play.
It absolutely should. And, thinking about how plants actively manage their own decline, how they prioritize recycling with such efficiency, and how some can even apparently maintain peak growth right up until a potentially sudden end. It really raises a fascinating final thought for us to consider. How might studying these precise, highly regulated, programmed processes of senescence and death in plants maybe change the way we think about fundamental concepts like aging, biological efficiency, resource management, and even the very definition of the end of life, perhaps even outside the plant kingdom itself?
A lot to chew on there. Definitely more going on in the life and death of plants than meets the eye. A truly deep dive.